Cellular respiration and photosynthesis. Cellular respiration happens in all animals. To make it occur, you need two reactants, glucose, which is ingested, and oxygen, which is inhaled. The products of this reaction are carbon dioxide and water. Tons of energy in the form of ATP is produced. The very first step of cellular respiration is glycolysis. This occurs within the cytosol of every cell. The glucose is broken down into two pyruvates. The glucose then has a phosphate put on carbon 6 to make it into glucose 6-phosphate using the enzyme hexokinase. ATP turns into ADP. The glucose is rearranged to fructose 6-phosphate. ATP becomes ADP and a phosphate is put on carbon 1, making fructose 1,6-diphosphate using the enzyme phosphofructokinase. Using the enzyme isomerase, the fructose 1,6-diphosphate is split into the isomers GAP and DHAP by removing the oxygen joining them. 2 NAD+ and phosphate turn into 2 NADH and 2H+. DHAP is flipped and there is a phosphate on both ends of the 3 carbon molecules. 2 ADP turns into 2 ATP as they take the phosphate group on the end of the pyruvate. The phosphates slip down to the middle and become pyruvate 2-phosphate. H2O leaves. 2 ADP becomes 2 ATP as the phosphates leave the 2 molecules, leaving behind 2 pyruvates and the end result of glycolysis. So the products of glycolysis are 2 ATP, 2 NADH, and 2 pyruvates. In order for pyruvate to enter the mitochondrion, it must be oxidized. This is what's called pyruvate oxidation. As the pyruvate passes through the mitochondrial membrane by transport enzymes, the carboxyl group in pyruvate is removed, releasing CO2. Then NAD+ is turned into NADH, and coenzyme A is added to make acetyl coenzyme A. The acetyl coenzyme can now enter the Krebs cycle. The 3-carbon molecule loses a CO2 and becomes a 2-carbon molecule called acetyl-CoA. Acetyl-CoA enters the Krebs cycle when it is joined to oxaloacetate by citrate synthase to produce citrate. Citrate is converted into isocitrate by the enzyme aconitase. CO2 comes out and isocitrate is then converted into alpha-ketoglutarate. NAD+ becomes NADH. CO2 leaves and alpha-ketoglutarate is then converted into succinyl-CoA by alpha-ketoglutarate dehydrogenase. NAD+ becomes NADH. Succinyl-CoA is then converted into succinate by succinyl-CoA synthase, which yields 1 ATP per succinyl-CoA. Succinate converts into fumarate by way of the enzyme succinate dehydrogenase, and FAD is reduced to FAD2. Fumarate is then converted to malate by hydration with the use of enzyme fumarase. Malate is converted into oxaloacetate by malate dehydrogenase, and NAD+ turns into NADH. Oxaloacetate is then joined by acetyl-CoA and the cycle continues again, hence the name Krebs cycle. The electron transport chain takes place in the inner membrane of the mitochondria. Dehydrogenase complex protein receives 2 electrons and hydrogen from NADH. Hydrogen moves to intermembrane space. It then passes the electrons on to the electron shuttle, ubiquinon. Ubiquinon shuttle carries electrons to second protein called cytochrome. FADH brings hydrogen to ubiquinon, which also takes them to cytochrome, and it passes through the intermembrane space. Electrons are passed to the second shuttle called plastocyanine. Plastocyanine brings electrons to the cytosome oxidase complex, where the electrons leave the protein and go into the mitochondrial matrix. The electrons enter the matrix when oxygen, the final electron acceptor, and 4 hydrogens form with the electrons to make water, which helps to keep the concentration. Hydrogen enters the mitochondrial matrix from the intermembrane space through the ATP synthase. 1 molecule of ATP is synthesized as it passes through the ATP synthase complex from the reservoir in the lumen back into the matrix. Photosynthesis. A plant captures CO2 released from animals through its stomata, and it absorbs water through its roots through osmosis. The first thing that happens is the electron transport chain. A plant captures CO2 inhaled from animals through its stomata, and it absorbs water through its roots through osmosis. The first thing that happens is the electron transport chain. 2 photons of light with wavelength 80 hit the antennae complex of photosystem 2. The photons bound around the active site. Water gets broken up into half an O2, 2 hydrogens, and 2 electrons. These 2 electrons now travel through the electron transport chain, replacing what leaves. The primary photon acceptor extracts the 2 electrons and moves the plastic quinine, PQ, to be transported to B6F. 4 hydrogen ions are dropped off in the lumen via the PQ. In B6F, the energy from the 2 electrons is used to make 4 hydrogens from the stroma to lumen. The 2 electrons from photosystem 2 are replaced. The electrons were waiting to be charged by a photon wavelength of 700. The 2 electrons provide energy to reduce NADPH using a hydrogen ion from the stroma. 2 NADP forms 2 NADPH. Hydrogen ions from the lumen pass through the ATP synthase to the mitochondrial matrix. The next process that occurs in photosynthesis is the Calvin cycle. This occurs in the stroma within the chloroplast surrounding the thylakoid membrane. The ATP and NADH from the light reactions act as fuel, driving this cycle. It is important to remember that the Calvin cycle is not dependent on light, just the products of the light reaction. The purpose of Calvin cycle is to produce G3P to make glucose. The first phase of photosynthesis is carbon fixation. In this phase, rubisco catalyzes 3 CO2s to react with RUBP and form 3 unstable 6-carbon molecules which immediately break down into 6 3-carbon molecules known as PGA. The next phase is the reduction reactions. 6 ATPs are turned into ADP so that 6 PGA are turned into 1,3-BPG. Then 6 NADPH are reduced to NADP, and the 1,3-BPG is turned into 6 G3P. One of these G3Ps exits the cycle as the final product. The third and final stage is the regeneration of RUBP. In this stage, the 5G3P, a 6-carbon molecule, must be turned into 3-RUBP, a 5-carbon molecule, which goes back into the cycle. First, inorganic phosphate is released. Then, H2O is used to rearrange the molecules. Next, ATP is turned over into ADP and 3-RUBP are left over. The G3P created as the final product of the Calvin cycle is turned into glucose, is used for plant growth. And eventually, this glucose is eaten by an animal to be used for cellular respiration.